My talk today is about biological oxygen activation. I will take examples from my research and show some of the results we have obtained. But first, I would like to show you a little bit about my career. I was candidate from University of Copenhagen in 1983 with my supervisor Erik Larsen. And when I finished my degree, I would really like to pursue a research career. And so I applied the Casper Foundation for a 3-year scholarship, and that was focused on bioinorganic chemistry. And I was so lucky to obtain that scholarship, and then I could be employed as a research assistant at the Department of Chemistry at KBL, where I had Erik Larsen and Erik Petersen as my supervisors. Erik Petersen was at the chemical department here at KU. I finished my licentiate degree in 1988 and was shortly after that appointed assistant professor at the chemistry department at KBL. And then I had another very great opportunity because the Casper Foundation was having a scholarship at Caltech. They call it the Casper Foundation Scholar at the Beckman Institute. And that was around 1 year of research money for a year at Caltech. I applied for that, and I was so lucky to obtain that scholarship, and I went to Caltech in 1990 for 16 months, where I was working with Harry Gray and Jay Winkler. And that really makes me sure that bioinorganic chemistry was a research field that I really enjoyed working with. I'm going to skip the next 20 years in my career. I became associated professor at KBL and finally full professor in 1902. In 2007, my KBL was joined together with the University of Copenhagen. We become part of science. And in 2011, the department at KBL was stopped, and all the people were moved around. I moved here to HCØ in 2012, together with Peter Tulstrup and Lars Hemmingsen. And we have been here now, and it was amazing to realize it's already 10 years now that I've been here at the chemistry department. What can I say? My teaching, I have at the moment 3 courses. I have a large course in general chemistry for students in food science and biotechnology. There's about 160 students for a first-year course. Then I help with a course called Bioinformatic 1, where I have some lectures. And then finally, I have a master course called The Chemistry of Metal Ions in Biological Systems, which is a way of teaching the chemistry I like to do for my own research. But now it's time to look at some of the results. And the first example I will mention is what happens when I was at Caltech. When I arrived, yes, but before I do that, I just have to mention a few things about dioxygen. The first thing about dioxygen is that if you take a molecular orbital diagram as shown here and count the 16 electrons in dioxygen, you will see that you end up with two electrons in the antibonding orbitals, the pi antibonding orbitals. That means that dioxygen is a diradical with 2 unpaired electrons. And due to Hund's rule, we have a triplet ground state. That actually makes dioxygen extremely slow reacting, because you have to do spin pairing when you are reacting with organic molecules. I've shown very schematically at the bottom here a reaction where a triplet state will react with a singlet state of some organic material, and eventually it should end up as a single state, as a diamagnetic compound. So we have to have a spin pairing, and that means that this reaction has a very high activation energy. And that's why dioxygen is reacting very slow. And that is, of course, lucky for us, because we have to control the dioxygen when we breathe. I have, for my teaching, developed a small experiment to show that actually it is paramagnetic dioxygen. It's a very simple experiment. I have a copper cone, and I fill it with liquid nitrogen. And because the boiling point is lower of dioxygen higher than liquid nitrogen, it will condense on the outer surface, and there will be droplets coming down here. And as you can see in the picture, this is a pure dioxygen, liquid dioxygen coming down. If I take a magnet, it's an old magnet from a hard drive, and get it closer, you can see that it is actually attracting the dioxygen. So dioxygen is truly a radical, a paramagnetic compound. Another thing we have to understand when we are discussing biology is that it's going on in water. And because of that, there are limitations for the redox potential that is available. The highest redox potential we can have in water without oxidizing water to dioxygen is around 0.8 of a volt. At the same time, the lowest potential we can have is about minus 0.4 of a volt, because else we will have hydrogen involved. So the only way to have higher or lower potential is that we have to have a system that is without water. But since most of the reactions are close in contact with water, this is a limitation we will have to have. And then oxygen has another problem. Oxygen itself is slow reacting, it's kinetically very slow, but as soon as we start to reduce dioxygen, we form a lot of species, normally called ROS, reactive oxygen species, which are reacting very fast. And they are a problem in biology, and biology has developed systems, as in our red blood cells and in the blood we have. There is produced a quite high concentration of superoxide, but we have an enzyme, superoxide dismutase, which is disproportionating superoxide. We have catalase that takes care of the formation of hydrogen peroxide. But we also have protein systems that can use dioxygen and hydrogen peroxide to produce important compounds in our living cells. But let's go and see what was I doing when I was at Caltech. I was very lucky when I arrived at Caltech. The Beckman Institute was just opened. I was one of the first there. And they have just installed a nanosecond laser that could help studying electron transfer reactions, because in biology, electron transfer reactions are very important, because normally electrons are moving one electron at a time in biology. So we were studying a system where we have a fixed distance between donor and acceptor, and we could initiate this using a laser. And then we could get an excited state, and we could measure the reaction back and measure the rate constant for that. We were using a very simple system. We use a ruthenium complex that was resonance. We have a protein, in this case, I showed here, a cytochrome C with an ion center. And then we were probing the surface with various histidines we were engineering into the protein. And then we could study the effect of distance, but we could also study the effect of the intervening media between the donor and acceptor. Some of the papers that has been published are shown down here at the bottom. And we learned two things from these experiments. the proteins is not like a glass where only distance is important. Proteins actually have pathways in the protein that can make a connection, a kind of wiring of the electron transfer. That means that the protein to a certain extent can control the way the electron is moving, and thereby avoiding that electrons is just moving arbitrarily by distance. It can actually make these kind of pathways. That was a very important thing when you talk about long-range electron transfer, that nature can control how they are moved around. Another thing we learned was that by changing the driving force, we could show that actually the inverted region can be obtained inside protein. So again, here there is a control. The rate in biology systems can be controlled by controlling the reorganization energy for the electron transfer. So these two things are actually quite important when studying oxygen activation in biological systems. The first example of a protein I would like to discuss is Laccase. Laccase is a multi-copper oxidase found in plants, but similar system is also found in our blood. We have a protein called ceruloplasmin, which is very much like a Laccase. At least it also has these 4 copper centers. Laccase is actually using typical phenols as substrate and oxygen, and then it can reduce the oxygen to water and at the same time oxidize the substrate. The 4 copper are sitting in some standard geometries. The T1 side is a special copper side which has 2 histidine and a cysteine. It is a copper side that is found in blue copper proteins because it has a very intense blue color, about 10,000 in molar absorbance. Then there is a T3 side, which is 2 copper that is bound antiparagnetically, so there is no EPR signal even if there are 2 copper 2 sitting in the T3 side. And then we have a normal copper, that is a copper where water is coordinating and with an absorption that is found as for the aqua copper 2 ion. How can these 4 copper make this kind of chemistry? The protein is extremely nice because it don't want to have intermediates coming out of the system. So there is this center here where the reduction of dioxygen actually occur is in the middle of the protein. There's a channel that allows dioxygen to move into this center, and there's another channel that allows water to leave the center. And then we have the substrate at the T1 side coming in, being oxidized. The copper is reduced, and it is sending by long-range electron transfer the electron into the trinuclear center. And after 4 reductions, we could have a completely reduced enzyme. We were doing some stop flow experiments where we were entering oxygen into the reduced Laccase. And the first step is actually the binding of a hydrogen peroxide, at the same time oxidizing the 2 copper in the trinuclear side, the T3 side. And then further on, the 2 reduced copper, the copper T1 side and the T2 side, are adding 2 more electrons, and we actually have an oxide coordinating to 3 copper. And then it can be reduced and protons can come in, and we have release of water. And so this is a catalytic cycle, very efficient. It's amazing you can have a color like reactive red or reactive black, very stable in water. As soon as you add Laccase, the color disappears. It is really a blue copper protein because this is a sample of the protein. You can see it has a very nice blue color. And this was done mainly at at KVL, but also some of the studies has been done together. We have done some X-ray structures, crystal structures, and this is together with Heidi Ernst and Signe Larsen. My next example will be LPMOs, which is a class of enzymes that is found in nature. And what they do is they are helping the degradation of biomaterial, of plant material, of cellulose. And why is that necessary? There's a lot of protein that can degrade cellulose, there's cellulases and so on. But the problem is a certain fraction of the cellulose found in nature, in biomass, is crystalline cellulose. And the crystalline cellulose cannot really be attacked by these cellulases because the structure is very compact. And these cellulases need loose chains of cellulose, because then they can put it into a channel and they can chop off these diglucose units, and then beta-glucosidase will make them back to glucose, and then we can make second generation bioethanol and so on. The structure here is a little odd because they can do this oxidation of the cellulose rings chain, oxidizing one chain of the one end of the chain to a acid. And that is a little odd how one copper can do this kind of oxygenase reaction. When I started into this field, people have developed a model for the reaction that it was dioxygen that was the substrate. It could react with a reduced LPMO, forming a superoxide radical bound to copper 2. And then by some kind of electron donation from other molecules, it could form this oxyl, copper 2 oxyl species that should be the active one that would take a hydrogen atom, and then it could put in back the a hydroxide group on the ring system, and then we would have a bond cleavage and we would have the acid formation. That sounds like a possible mechanism, but the problem was that it was very hard to figure out where the electron was coming from in this system. So I entered into this because I thought maybe there was some step involving copper 3, so I was looking at that, trying to see if I could identify an intermediate of a copper 3. Then I come in contact with Klaus Stelby, who was also working on these LPMOs, and we discussed if it was possible to inject electrons into the system using light. We were using chlorophyllene, which can be excited, and then in the excited state, it can inject an electron somewhere. We were hoping into the LPMO, and then we could reduce the oxidized chlorophyllene back to chlorophyllene, and then we could just continue using light as a source of electrons. And eventually we were using ascorbic acid, but we were also showing that lignin, for example, could donate electrons to this system. But it was very odd. We were doing time-resolved fluorescence measurements, and we could not see any interaction between the chlorophyllene and LPMO. So we were ruling out the possibility of interaction here. So this direct electron transfer was not likely. At the same time, we were observing that we had a high production of hydrogen peroxide. At that time, when we were not really finishing and we were not really strong believers what was going on, there was a paper by Bizarro and from a Norwegian group where they proposed that hydrogen peroxide was actually the real substrate for these LPMOs. And that is, of course, a reasonable approach, because dioxygen, 2 protons and 2 electrons, is actually the same as a hydrogen peroxide. So hydrogen peroxide will deliver everything needed to perform this reaction. And why people were not believing that hydrogen peroxide was a substrate was actually due to people had been doing these experiments, but they were inactivating the LPMO very fast when they were doing these experiments. So what happened was that they actually was doing quite odd experiments, but they had some reason to believe that hydrogen peroxide could work. We took up this because we knew that in light we were also forming hydrogen peroxide, and when we were using light, we actually accelerated this reaction here by 20 times fold. So if hydrogen peroxide was the substrate, then this reaction here was just generating hydrogen peroxide that could be used by the LPMO. So it was not, then we could rule out the direct electron transfer, but we knew that this system actually activated the enzyme by 20 times compared to normal oxygen reactions. So in order to have stoichiometric or substoichiometric amount of hydrogen peroxide, we use very, very large amount of LPMO. You can see 1 millimolar. Then we could add 1/5 of that in hydrogen peroxide. That means that only 1 hydrogen peroxide per LPMO, actually only 1 per 5 LPMO. And then we saw the development of an intermediate, very long-lived. You can see the decay rate was something like 10 minutes. And that was interesting. What was this species that was formed with the direct interaction between LPMO and hydrogen peroxide without substrate? We were studying that and using resonance Raman, EPR, and so on. And we were able to show that what actually was formed was a tyrosine radical, a copper-2 tyrosine radical. And that was fine because there was this tyrosine in the structure, as I showed here. This tyrosine is sitting actually in a very nice position for making this kind of interaction. In the beginning, we were looking if this was the substrate, but the mechanism now in the literature is mainly that it is the oxyl radical that is active. But if there is no substrate present, this is so reactive it would oxidize the protein itself. But forming this long-lived intermediate, then it could protect itself. And if there's no substrate present, what we see is a slow oxidation of tyrosines on the surface of the protein, forming dityrosine. So the enzyme is still active even if there is no substrate. So even if it become oxidized, it can survive this oxidation without being destroyed. If 2 hydrogen peroxides enter the protein, it's completely oxidized. So that's why it's only when we apply hydrogen peroxide in a very low doses on a continuous base, like in our system here with the light, that we can accelerate the reaction with hydrogen peroxide. So it's a very delicate system, but in nature, there is a production of hydrogen peroxide all the time. So it is reasonable to believe that both dioxygen is the substrate under certain conditions, but also that hydrogen peroxide can be a substrate under other conditions. This project was funded by a Novo Nordisk grant, and we were a large group. We could do a lot of different experiments, but this is just one example of the results we obtained. My last example of my research is metal-catalyzed protein oxidation. And in protein oxidation, we have this, that dioxygen is slow reacting. So normally it's not a huge problem. But if metal ions are found in the system, they can catalyze this reaction dramatically. And so we were studying oxidation of various proteins using a little bit of copper ions and a little bit of hydrogen peroxide. We were looking at the formation of oxidized proteins, and then we were using a lot of different techniques to figure out what was going on, which amino acid was oxidized, and so on. The funding here was a DFT grant, where we in 2015 got a big grant, so we could do a lot of collaborations. The reaction with copper, reduced copper and hydrogen peroxide, is that you can form this hydroxide radical in a kind of copper-catalyzed pentane reaction. And this hydroxide radical is extremely reactive, and it survived very shortly in a biological system, less than a nanosecond. So it's oxidizing always very close to the production side. And it's extremely reactive. It can react with all types of amino acids. And so we were looking for some of these oxidation products, dityrosines, 2-oxohistidines, and so on. And we were having 4 different systems. We were having a system from that is important in Alzheimer's, Parkinson's disease. It's a protein that forms this plaque in the brain that is part of the Parkinson's disease. And we were looking if this could be accelerated by oxidation if there were presence of metal ions. We were using a copper zinc superoxide dismutase to see if there was special protection mechanism for an enzyme that was dealing with a lot of radicals. We were also looking at toxins, if we could detoxify some toxins using this method here, and then we could make vaccines. Actually, we succeeded in making a vaccine that worked on mouse, but that is another story. The example I will tell you about is the superoxide dismutase, which is an enzyme that is disproportionate superoxide ions very, very fast. And the idea is that we have a copper that is active, sitting in a cavity, and it is also controlled by a structural zinc that is holding a bridge of a imidazole ring between the zinc and the copper. So it's a kind of ping-pong mechanism. The first thing that happened is that a superoxide coming in, and we reduce the copper 2 to copper 1, we break the bond to the imidazole, a proton can enter, and when the next superoxide coming in, there is both an electron and a proton ready, so it can be reduced to a hydroperoxide ion, and then when it goes into solution, it can take another proton and become hydrogen peroxide in solution. When we took this enzyme and looked at it at our beautiful infrastructure in the mass cellar we have here at the department, we could see that most of it was intact protein. We had a little bit of oxidation in our protein, not much. But as soon as we exposed it to micromolar concentration of copper and hydrogen peroxide, 140 micromolar copper and around 500 micromolar hydrogen peroxide, then within seconds, we start to have a strong oxidation. We could stop the reaction with EDTA. It completely stopped the reaction with 2 millimolar EDTA. So we could stop it at different timescales, and we could follow what happens to the proteins when they were oxidized. And using tryptic digestion and mass spectra of the peptides and so on, we could identify the sites of oxidation. And here I will just mention 2 things. The first thing was that methionine was oxidized very fast, and that is not influencing the activity at all of the protein, of the enzyme, the SOD. But then we saw also that the disulfur bridge was oxidized. actually we could identify this intact disulfur bridge that was oxidized at one of the sulfurs. That means that we have two groups that can take oxidation hits without destroying the function and the structure of this enzyme. So it is protected. And then when we continue to oxidize, then we start oxidizing the histidines, and that of course would kill the activity completely of the protein. But in nature, the conditions are milder than the one we were using. So it is likely that these enzymes can take some hit and still be fully functional. That's a clever strategy, and that was, I said, especially this intact oxidized disulfide bridge, I think is very interesting, because that could be something that happens a lot in these systems. And with that, I would just say thank you for your attention.